Hello, my name is Praneeth. I worked under the mentorship of Dr. Hunter Mosley at the Mosley Bioinformatics Lab at the University of Kentucky, and my project was titled Omics Error Analysis and Visualization. So to give you a brief overview of what this presentation will consist of, first I'll begin with an introduction, then I'll go into the goal of my project, I'll discuss how I did it in my methodology, then I'll discuss the data I created, then I'll go into discussion about this data and make some conclusions. Then I'll discuss some further steps in research that I can do. Then I have some people I would like to acknowledge. And then I'll go into some references and then open the floor for some questions. So to begin with, what is omics? So omics refers to the systematic study of molecular entities in a biological system. So basically, it's looking at really small things within your body. And this can be divided into a whole bunch of different categories. If you want to look at DNA, then you're looking into genomics. If you're looking into RNA, transcriptomics, proteins, proteomics, and so on. There's a whole bunch of different subsections of this. But in all of these studies, you're using technologies and generating lots of data. And so you have these really large data sets, and it's natural to assume that these data sets will contain error because all of these technologies that you're using to gather this data isn't perfect. There could be any slew of errors within the detection itself, with sensor problems or anything that could cause error to occur and for your data to not be completely accurate. So it's healthy to assume that error exists, and so there's two main types that we're looking at here. The first one is constant error. Basically, across the board, there's a consistent amount of error throughout as your mean increases, decreases, whatever, the amount of error is constant. The other type is proportional error. That means as your mean increases, for example, your error also increases. So the more you have of it, the larger your error got. It wasn't consistent throughout. So with all these data sets, right, there are large public repositories where these are stored. Namely, the two main ones are the Sequence Read Archive, which is the primary archive for high throughput sequencing data, and the Gene Expression Omnibus, which focuses on high throughput functional genomics data. So specifically, genomics is what that one's focused on. And then the all RNA sequencing and chip sequencing sample and signature search, or ArchES4 database. Essentially what they did was it mined the SRA and the GEO for RNA sequencing data from humans and mice specifically, and put it into a format that could be accessed by programs. So the goal of my project was to develop methods to automatically evaluate the amount and type of error in every data set in the SRA, and this extends also to the GEO. So how I went about doing this was, first I obtained a set of samples that I could use from the ArchES4 database. If you'll recall, they compiled all of these data sets, so I took a few samples from that I could use to test my program. Next, I created a Python script that essentially took all of the data sets, the samples, and separated them out, and found all the replicates using the sample title. So how I did that was, there's this really cool thing called Levenshtein distance. And it means the amount of single character edits it takes to change one word into another. For example, if you want to change cat into dog, it takes 3 single character edits to do that. You're changing the C to the D, A to O, and T to G to get from cat to dog. And so the Levenshtein distance between those two words is 3. And so I was able to use this to look at all the sample titles, and then I could calculate the Levenshtein distance between all of them and say, and group all of the samples with a title that had a legacy distance less than 3, because basically what that was doing is it was grouping all the replicate samples together. And so that way I could make statistical calculations based on that. So I was calculating the mean, the standard deviation, the relative standard deviation, and the difference in range between all of these replicate samples. And so after that, I plotted all of this. So first, this is when we're first looking at our data, this is first thing I did was I created a simulated data set. So I didn't immediately go into my samples. I first made my own random number generated data set, and I performed these same statistical calculations on them. And then I graphed each of the three, the difference in range, the standard deviation, and the relative standard deviation against the mean. And so how I went about doing this was I made 3 separate data sets. One where only constant error or additive error was introduced into the set, meaning the error was constant throughout the whole thing. So only that type of error was in that set. In the second set, I introduced only proportional error. So I made sure there was no constant error, only proportional error. I manually added in this error. And in the last set, I made sure I added a mix of both to see what would happen. And so when you're looking at the constant error graphs, you'll see the most important one to notice is the very last one, where you're graphing the relative standard deviation versus the mean. You see a very strong inverse exponential curve pattern you see, right? And then if you're looking at the one with only proportional error, you'll see that for when graphing the difference versus the mean and standard deviation versus the mean, you see very like y equals x like graphs where it's just a linear line. And so what happened when you graphed a set with mixed error is that both of these trends actually continued. So in the mixed error, you see those linear lines that occurred when there was only proportional error, but you also see that inverse exponential like graph from the constant error. And so what that showed to me was I could look at a graph. If I took a data set and graphed these, I could look at it and know what type of error I'm dealing with. Because I can look for just this one, I know looking at it because it showed the same graphs and patterns as the ones when there was only each type of error, and it's showing both of them. I know that it has both types of error. And so the next thing I did was I did all of this, and I ran the plots onto the sample sets. And so one example of this is the GSE 52934 sample data set. And so I ran all the calculations on this set, and then I plotted them. And you'll see immediately it looks very similar to the simulated set with mixed error. You see, again, the 2 linear like graphs, and then followed by an inverse exponential curve. And so basically this shows to me that this set contains both constant and proportional error. And so that's really important. And so basically, so that means I was able to accomplish visualization of these graphs. And so I can show the effect of error on these plots because of the simulated set showed what the error looks like, and then when you use it on actual data sets, and I can still see those similar trends, that shows me that I can use this to identify those types of error within those graphs. So again, if you compare, if I go back and forth quickly between these two, you see the graphs look very similar. So some further steps of research that I can do is currently I'm using a library called dockup to create a command line interface that allows basically anyone to use this script very easily. And so allowing command line options makes it much more robust and usable, especially since the goal here is to make a script that anyone can use and become publicly available. Secondly, I'd like to add more visually informative plots. Right now, all the graphs don't tell anything by themselves without having prior knowledge about this. I would like to add information visually that explains already what I'm thinking and tells people that don't already know about it. And then finally, what I'm working on right now actually is taking those statistical calculations that I've already made, and for each of those sample sets, calculating the actual amount of each type of error. So obviously visualization is really important because it'll tell us how much error, not necessarily how much error there is, but that there is both types of error or one type of error in there. And so that's really important to account for, knowing that you have error. But then the second step of that is to actually know how much error you have, because if you want to use these data sets and you know there's error in it, you want to know how much of that error there is so you can account for it later. And so that's what I'm working on right now, is to give numbers specifically on how much of each type of error there is. So I'd like to quickly acknowledge Dr. Hunter Mosley and Dr. Robert Flight. When I joined the Mosley Bioinformatics Lab, I knew very little. They taught me a lot, starting from just the Linux OS to virtual machines to learning Python from scratch. And I was able to do all of this thanks to them. I'd like to thank the Kentucky Young Researchers Program, MSTC, and Ms. Karen Young for allowing this opportunity for me to do research in a high school setting. And I'd like to thank my family and friends for being so supportive over the last 2 years. These are my references, and if you have any questions, feel free to email me. Thank you.